I am a program manager at the National Center for Advancing Translational Sciences, or NCATS, which is one of the institution centers at the NIH. I'll talk a little bit about what that means, but I'm going to talk to you today about two different programs that I am heading up. One called Biomedical Data Translator and the other called LitCoin. So what's NCATS? NCATS is a disease agnostic branch of the NIH. And what that means is a lot of the other institutes and centers at NIH focus on a specific body system or type of disease. So NCI focuses on cancer, and HLBI focuses on heart, lung, and blood. But NCATS is different in that we don't have a disease focus. We have a more generalized focus on translational science as a whole. So what that means is that we generally look at anything, and fund researching any field that would be relevant to translational science. What it also means, though, is that we're small, very small compared to some of the large institutes like NCI. And so we have a small budget, and we also don't have an R01 research program at all. So all of the grants and proper agreements and things like that NCATS funds are generally focused on short periods of time, relatively small amounts of money to support research that will hopefully lead to something like an R01 with one of the institutes or a large grant from another funder. But it also means that we get to focus on very innovative funding opportunities. And so I'm going to talk to you today about two projects that have very innovative funding structures. And I'm very excited to be working on those and not working on just a large cadre of R01s like a lot of other programs we have. And NCATS does have an emphasis, though, on rare disease research. So we focus on rare disease because a lot of the rarest disorders are not well funded within the research spaces that they normally. So rare cancers are funded on a much smaller scale than lung cancer and things like that. So we do have some rare disease research. And within NCATS, I am in the Office of Drug Development Partnership Programs. And so all of the work that I do, I sort of start from the direction of can we create a system that will help get more drugs from basic science bench level to the clinic more quickly. I am a program director for the Biomedical Data Translator, and within that project, we have 14 funded teams, and so my entire award portfolio that I work on at NCATS consists of the 14 teams that are working on Biomedical Data Translator and the few awards that we've given for LitCoin so far. I am in charge of the scientific oversight for these programs. I review the funding requests for each of the teams that are funded by us and approve their budgets. I also review and approve their milestones for each funding year. But the majority of my time is taken up by attending working groups and committee meetings for the translator system, translator program. And so then that helps me lead the decision making on program priorities for the program. But as I said, I'll also talk a little bit about LitCoin. And so with LitCoin, we've run 2 challenge prizes to build the initial design of the LitCoin program. And then also we have a contract now in place to build the foundational knowledge graph that I'll show a lot more about here in a few minutes. So for the Biomedical Data Translator, this is the problem we're trying to solve. And we like to call this the chasm of semantic despair. And what that means is basic scientists and clinical researchers and clinicians really, they talk about disease in a fundamentally different way. Basic scientists talk about, the underlying genetics, the underlying proteomics that lead to a disease, the pathways that might lead to a given disease phenotype, whereas the clinicians really look at signs and symptoms in the lab or in the clinic, and how they can affect the symptoms that patients have. So our goal with Translator was to try to bridge this gap by creating a software system that includes data from all these different walks of life within biomedical research. So what is data translator? As a whole, Translator is meant to pull together many disparate public data sets into a large cloud of connected data, as you can see here. And the goal of the actual, so we've done that. We've pulled together hundreds of databases and connected them in ways that I'll explain to you in a few minutes here. But what the software system that we've built for Translator actually is going to do is to explore pathways through that very complex cloud of data and try to answer a query for the user. So your query might be something like, what drugs might treat Alzheimer's? And we could return pathways through our very large cloud of data that might lead to potential treatments for Alzheimer's. So this is the architecture of the Translator program. And these nodes within this pseudo graph here represent different teams within our consortium, as well as the public databases that they're accessing. So at the sort of lowest level of the architecture, we have the public databases and knowledge sources. So all the data is accessible out there in the world already. And we have knowledge provider teams whose job is within a specific data domain to reach out and find all of the important data and knowledge within that domain and try to conglomerate those into knowledge graphs that we can then access. So for example, we have a genomic data provider team, clinical data provider team, chemical data provider team, and several others. Each of them within their domain of biomedical research is meant to pull together large graphs of all of the relevant data. Built on top of that, we have the autonomous relay agent teams, and those are essentially the reasoning agents within our system. And so we have five of these AROs in the consortium right now, and their goal is to accept a query from our autonomous relay system. I'll talk about in a second. They accept a query. They analyze and interpret that query, and then reach out to different knowledge providers to say, I need this data from the chemical provider, I need this data from the clinical provider, et cetera, and pull that data together into a relatively small knowledge graph that represents the information that could be relevant to the user's query. And then they make a path through that data. So they make multiple paths through that data. And those paths represent potential results to the user's query. And then they present them back to the autonomous relay system, which is the ARS, or we call the ARS. That is the system, I'm sorry. The autonomous relay system is the piece that's being built by NCATS ourselves. So one interesting thing about our program that is very uncommon is that we have extramural researchers being funded by the program, 14 different teams across the country and abroad. And we also have an intramural research team who's building that autonomous relay system part of this the system. So we have collaboration between extramural and intramural researchers. So that autonomous relay system, their job is to serve up those results that came from the different arrays to our user interface and try to give the user the most relevant results based on their curve at the top. And I'll show you how that looks in a few moments here. And so this is the flow diagram of the runtime of a translator query. And I'm showing you this mostly to show you how complex it is. So all of the arrows that are being shown here represent some information that needs to be transferred between software systems within the Translator consortium at the level of a single query that comes in. And so, for example, you notice I have this just represents KPs here. This node here represents all of the knowledge graphs that we've already built within the system. And everything else is the accession of those knowledge graphs. So again, it's a very complex system, both at runtime and in the case of building the graphs themselves. One of the ways that we have made this system work is by creating an API that we call TRAC, the Translator User API. And this API standard allows us to pass graphs, node edge node graphs between different software tools, all within a singular format. And so we represent queries, graphs that come from these queries, and results themselves that are being returned to the UI, all using the Trappy standard. So it's JSON based. It has been developed entirely by the Translator consortium over the past 7 years. And it is composed of a query graph that represents the user's question, a knowledge graph that represents all of the information that was relevant to that question according to one or more of our arrays. And that holds the nodes and edges that define that information. And then the results set that is a subset of the nodes and edges that are in the knowledge graph. Now, this is a representation of the ontological framework that we use for building all these knowledge graphs. So one of the biggest issues we've run into, of course, with this large amount of data is this data is very disparate. It's in different formats and in different domains. And so we need an ontological system that can represent all of this very disparate data across all of these different fields. And so this is the entity hierarchy of the BioLink ontological system. What that means is that we have all of the different types of entities, all the different types of nodes that we model within our system are represented somewhere on this graph. And you can see it can go from things like microRNAs back to RNA products, back to transcripts, all the way back to named thing, which is the most generalized concept in this model. And what this allows us to do is that when our translator tools are querying the underlying knowledge graphs, one of them might ask for example, an RNA product. But because of this hierarchical model, the returned results might include, instead of an RNA product, it might include something that's considered a microRNA, right? It's not just the one type of data that is available, but also all of the hierarchical underlying data types are also available and can all be returned given a specific query. And now this Sankey diagram represents, and this is actually a couple years old at this point, probably have another 50 knowledge sources, but this represents the knowledge sources that we have had access to with Translator. So all the way on, all of these on the left side are names of public databases, essentially. And so in this subset of them, you can see here that we've got things like Drug Bank, we've got the wellness database from the Institute for System Biology, we've got Kimball, we've got OMIM, all these different public databases that we have access through to in Translator. And the little colored edges here represent which of the funded teams actually use that data. So the connection site hypothesis provider, and they use some database that's up here further. Molecular data provider has access to all of these databases that are here in purple. And so you can see that, OMIM might be used by clinical data services provider, and they also might use Drug Bank. But then OMIM is also available through the multi-omics provider. And usually that means that they're accessing different types of data from that underlying database. And so then they have several other that I'm pointing here. The way that we keep all these different tools straight and allow them to interact with each other is by having them all registered in what's called a Smart API registry. So there's a registry of all the Translator tools all the software tools that we have within the Translator. It uses the OpenAPI version 3 structure. And it provides annotated metadata about where, the URLs where all of the different Translator tools can be found, what they can do, what data they have access to, and how those data are connected together. So we have not only, what is it and where is it, but also how are the different data points connected within a given graph for one of the knowledge providers. And so that means that it's entirely parsable by software. So going back to the architecture, in addition to all these funded teams that we've had around for several years now, we also now have a user interface team. So that's being built by a contract that was given to a company called BMA. And they are building the user interface for Translator that will actually allow our users to submit queries, that represent the questions that they have, the biomedical questions that they have, and they want to get answers to. And then the user interface translates their query into a Trappy, Translator API format, and then sends it over into the autonomous relay system to go and access all of the different knowledge sources. And then the results that come back are then sent back to the user interface, where those knowledge or those results are displayed to the user. So this is the timeline of the program, and you'll notice that there was a feasibility phase that happened a few years ago, but right now we're in the development phase of Translator. So this is, we've basically been working on the full development of the system for 4 years now. We're just starting our 5th year of development. And the user interface contract has been around for 2 years, and we're starting the 3rd year of that contract now. And we've recently had our initial public release of the Translator system. So it is available publicly. You'll see the URL at the end of the presentation. It is available to try right now. And hopefully I'll go through a couple of examples here and you'll understand how to use the system before you actually go into that at servers. So, let me go back here. So you'll notice that there are all these little lines here, and these lines represent all of the meetings that we've been holding for Translator. So we hold consortium-wide meetings, generally 3 times a year. And in the recent months, we've actually been able to do in-person meetings again, which we hadn't been able to do for in fact, the entirety of the beginning of the development phase with all Zoom-only meetings, of course, because we're in the middle of COVID. But we've been able to start having our in-person meetings again. And so we hold these meetings, in-person meetings 2 to 3 times a year. These are working meetings where we expect everyone to be actively hacking on the software. In fact, we used to call them hackathons, but NIH decided they don't like the term hackathon anymore, and they want us to call them relay meetings. We do have the in-person component wherever we can, and usually we have people, groups within the consortium working very late into the night on this work, and it's extremely collaborative. And so you'll see on the top right here, that is Will Bird, one of our funded researchers, showing us something. I think this was at about 12:30 in the morning at the end of the first day of our meeting. He had already been working for several hours on a plugin for ChatGPT that would connect to Translator and let him ask Translator questions using ChatGPT as the front end. And he then presented that to us the next day at about 1 o'clock in the afternoon. So he had written that overnight completely. So who are we building Translator for? The system is meant to be for translational researchers. And so we want to support people who are looking for support for their lab-based observations and trying to come up with new hypotheses that they can continue testing on. We think that in general, early career researchers and trainees are going to be very excited about this, and we think they are our ideal initial user base for Translator. But also we have another group of users that are using Translator every single day, or at least once a week. And that's the Precision Medicine Institute at University of Alabama, Birmingham. They have a group of clinician scientists who are using Translator to try to find new potential treatments for patients with very rare or very refractory conditions. And they use Translator weekly in their meetings to try to find potential treatments that they can give to these patients' clinicians, and the clinicians can potentially try to treat these rare diseases. And then we also are building Translator with the expectation that we'll have data scientist collaborators as well, who will want to both contribute graphs to the Translator ecosystem and also make use of all of the knowledge graphs that we've built. And so that's part of why we have a very strict API that we're using for all the knowledge graph transference between the different tools, because we want to make sure that anyone that wants to access our graphs Once they've learned how to structure a trappy query, they can make those queries, get back results, and utilize the graphs. So during this initial sort of alpha release period, we have focused on a few specific types of queries that we're supporting. And one of the reasons that we did that is that the biomedical knowledge space is very large. And so we wanted to make sure that we chose queries that we knew that we could support very well, answer the questions very well, and make sure that all of the results are supported by the provenance and evidence that comes from our graphs. And so those questions are represented here. The first one being, what drugs or chemicals may treat your disease of interest? What genes might be up or down regulated by a drug or chemical of your interest? And then what drugs or chemicals may up or down regulate your gene of interest? So those are our three queries that we support in our user interface right now. What this means is that we've had to come up with some really complex and difficult scoring metrics, because the goal is to try to come up with scoring metrics for the results that are coming back that are essentially universal across all of the queries that we support. So right now we use a combination of the scores that each of the separate reasoning agents are returning with a given result. So if multiple of our reasoning agent teams or reasoning agent software tools return the same result, then that result gets a higher score. If it only came from one ARA, it might be, the score itself might be based on the evidence that was used to generate it, for example. And so, of course, higher scores are there if there's more direct evidence for a result. And if there's predictions software predictions that our tools have been using, those also contribute to the score, but in a smaller way than direct evidence from our knowledge graphs. So this is the Translator user interface, and I'm going to just walk you through real quickly what running a query in the user interface would look like. So we're going to choose the genes to chemicals query to start off, and we're going to say we want to find a chemical that regulates a particular gene and that is going to be upregulated. And so I'm going to use a gene from my graduate school work, which is called FRAS1. When I was in grad school, I studied the mouse models of congenital diaphragmatic hernia, which is a hole in diaphragm muscle during development. And this is one of the genes that is implicated in the both mice and humans who have that disease. And so, once you hit search, then it's going to, while it's doing all of its work in the background, it's going to show you a little bit about the query, about the things you query on. In this case, a description of what the FRAS1 gene is. And this is going to show you a set of results. And so these represent different potential chemicals that might upregulate the FRAS1 gene with very little contextual information on the front of this. So this might be in any given body system, or it could be in a rat or a mouse or anything like that. This is a very general query of what could increase FRAS1.
What's the total population of drugs that is screening?
Total population of chemicals that is screened. That is a good question. A lot. I don't have an idea for you.
Tens of thousands, hundreds of thousands.
I would say in the hundreds of thousands. Yeah, definitely. Yes.
There's a question from the chat.
Okay.
From David Ashburn. Does the alpha query take into account species, tissue, developmental stage? I can see that there are opposite answers depending upon like tissue and age.
Right. So the answer is right now, no. We don't take with the high level query, we don't take those things into account, the age or the tissue type or anything like that. Our goal right now is to present to the user all of the potential things that might upregulate the gene, and then give them access to the underlying knowledge representation where we got that. And so the goal is give them all the possibilities and very quickly show them what the context was by sending them to the paper or the database or wherever it was that it came from. Our longer term goal is to start including those contextual relationships up front, maybe as filters, for example. So you might say, I don't care about anything that was done in mice. I only want to see clinical trial information. So you could filter for only results that include a clinical trial as one of the result editors, for example. Yes.
Is this like species specific or like, is this a filter you can do at the beginning or it's just like any species?
So right now, so you can definitely filter by species. The query that I made here was specifically on the human FRAS1 gene. So you can search for mouse FRAS1 as your starting gene. So that is one place where you can specify a species. But these results will not be only human specific right now. We will need to build a filter in to allow, again, if you really only want information on what was done in humans already, then you're gonna have to filter out the mouse and rat studies and things like that. Okay, so this is what the first few results would look like for that query. And if you expand one of these, in this case valproic acid, you'll see a very simple graph that represents the relationships between valproic acid and FRAS1 gene. And in this case, it's saying that there's a relationship between valproic acid and FRAS1 based on the FGF10 gene. So we can look at that in a little bit more detail. And so in this case, it says that valproic acid is known to cause an increase in FGF10, which is known to cause an increase in FRAS1. So we have a sort of two-step relationship there. You could use valproic acid to increase to upregulate an upregulator of FRAS1. And so if you click on one of these green edges here in the middle, it'll show you the actual evidence that we have to support this. So in this case, it's a set of publications that support the relationship of valproic acid increasing the expression of FRAS1. And if you were to click on this little read more button here next to one of these, so if you click on the little read more button, that would take you directly to the PubMed page for that paper. But of course, we don't just have publications information, we have other sources. And so if you go to the sources tab here, it would show you that the comparative toxicogenomics database has an entry that suggests that valproic acid causes increased expression of FRAS1. And you can click on the CTD link here and it'll take you to the CTD web page. Now, one of our goals in the longer term is to have this link right here take you directly to the database entry that supports that assertion. Right now, this takes you to the front page of CTD. But our goal is to try to get you to the specific database entry that'll support the assertion that is being shown. That's going to, hopefully, I think we can probably get there within the next year, but it's going to be a lot of work to find the URL structures of all these different databases and try to build those in. And so that's sorry, and this is a wiki page explaining what CTD is, and then it takes you can click on the CTD homepage there. And this is another one of the examples of, this is a search for what drugs may treat the rare disease epitheliomyopathy. You can see that this is a much more complex graph that was returned, and there are several different pathways through different sets of genes that support the possibility that this collagenase that was returned may treat epitheliomyopathy. So this, I just want to really show you that you don't just get very simple, one or two hot graphs. You get potentially very complex graphs that support a given potential treatment. And so if you, for example, were interested in the collagen 1A1 gene, you can click on that and it will show you only the paths that include collagen 1A1. Although it still highlights this one in the middle. I told them that they would change that. I think they changed that at this point. But the goal here is that if you know that collagen 1A1 is an interesting pathway for you, you could highlight those. So I mentioned earlier that the Precision Medicine Institute at UNB uses Translator all the time. And so this is sort of talking about how that's done. So they have a group of very, very active clinicians who are bringing their rare disease patients to Precision Medicine Institute all the time. And they're basically using Translator, mining the data to try to find potential drugs that might affect one of these very rare cases. And so this is one example. It was for a very rare syndrome called Shine syndrome. I think right now there are only 5 patients represented in the world that have Shine syndrome. And so when you have a very small, very rare disease with a very small number of patients, you generally can very easily find out what gene is associated with it. And so in this case, we know that the DLG4 gene is the cause of, mutations in the DLG4 gene are the cause of this disorder. And so it's characterized by sleep disturbances, hypotonia, intellectual disabilities, neurological disorders, and epilepsy. And it is predicted to be based on variants that are loss of function or haploinsufficiency of the DLG4 gene. And so in order to try to find a potential treatment for these patients, they started by asking Translator what might upregulate the DLG4 gene, because it is caused by haploinsufficiency. And so at the beginning, Translator tools really returned a ton of potential treatments. And then they had their analysts go through all those potential treatments and write a report of the potential treatments that are already available to a clinician that could have an impact on the expression of DLG4. And so one of them they came up with was guanfacine, which is a common blood pressure treatment. And so in this case, they gave that common blood pressure treatment to a little girl who has Shine syndrome. And after a few months, she was seeing significant improvement in her motor skills and her social and behavioral skills. And so I can actually show you that. So her mother had sent pictures into the UAB team of what her coloring books looked like. And just after, I believe this was a 4-month period, her mother said she had a real improvement in her Peppa Pigs. And so you can see, coloring in the lines as a motor skill measure to this mother meant everything, right? And so we've actually had clinical trials that have resulted from both this example and many of our other cases. And so this really shows that Translator can give value at all stages of research, from early preclinical all the way to direct clinical relevancy. And so this is just sort of talking about how, of course, we can't ignore the fact that LLMs are now a thing, right? And so we have these incredibly dense models, language models that are revolutionizing a lot of our natural language processing and things like that. And so we are in the process now of looking at different ways that ChatGPT and other LLMs could be valuable to us in Translator. So one of the things that we're definitely going to be looking into is, can we use an LLM to take a person's query in natural language and translate it directly into a traffic query that can be sent to our system? That would be one obvious good use of LLMs. But another one is shown here at the bottom, which is, when we get back a result, we usually have a large list of publications that support different parts of that result. And so can we ask a large language model to review all those publications and summarize the information that we return to give you a good high-level summary of, why a given result might be valuable to that user? And so that's another way that we're looking into using LLMs. So at the moment, that's where we're at. And what's next? We want to support additional templated queries. We're working on new templates now that we're going to start to build in the system. Eventually, we want to be able to support the very generalized query. What is the relationship between concept 1 and concept 2? And find us valuable points between those two concepts. We're still, a while away from that general type of query, but we're definitely making headway towards that. We definitely also want to make performance improvements to Translator. So right now, you usually get to see results from Translator within about 15 to 20 seconds. And then as additional results come in from our different tools, it will update those and tell you new results available, click here to refresh. So normally we have all of the results that are available within Translator in about 2 to 3 minutes. We want to shrink that down a little bit and get it down to the less than a minute mark. And I think we definitely have the ability to do that. We also want to support more concurrent users. We don't actually have even an estimate of how many users we can support right now, because we haven't gotten enough users concurrently to see any major slowdowns. So that's one thing that we're going to be watching really closely over the next few months as we put the URL out there in more public spaces. Is how many users does it take to start slowing things down, to cause one of our tools to potentially crash, and how do we then harden those systems against those potential crashes? And then we have a lot of documentation improvements that we want to make, including tutorials for collaborators that want to join in and contribute data and utilize the translator knowledge graphs, and also user guides for users of the user interface to understand what translator can help them do. We also want to allow users to bring their own data into the ecosystem. And what that means is that there are cases where you have data that you think would be valuable to the queries that you might have for translator, but you can't share that data. And so this might be, for example, companies that have proprietary data about drugs. It might be academics who have pre-published data that they want to, they know that that pre-published data can be really valuable to the algorithms in the background of the translator, but they can't share it directly. And so we want to make sure that we can support that by creating, and sort of the process right now of creating a sort of I don't know, a place where you can go in and deep dive into the data a bit more. Hopefully, in the next year or so, where we also allow you to upload your data in a way that it does not get put into our public spaces, but then their algorithms can have access to it and can make new connections between what we already have in all of our knowledge graphs and the knowledge issue update or upload. And then we're also working right now on what's called pathway figure OCR. And so the idea there is that, oh yes, go ahead.
What is uploading our own data? Is there any kind of format for this one, or you can accept any kind of data from multiple investigators?
Yeah, that's a good question. So as far as the formats of data that we can accept, they're pretty wide ranging. The way that we do that is one of our funded teams has really spent a lot of time on creating APIs that can access data and translate it into the TRAPI or the knowledge graph format that we use. So we will have to automate that in a way that you can upload what even kind of different types of file formats these might be, but it's gonna be relatively restricted to start, and then we'll hopefully branch out and allow for more and more types of data formats. But the important part is that the information you're uploading is not just numbers and values, but it has to be relationships between objects. So telling us that gene A has an expression of 15 in this case in your data doesn't tell us anything. But if that is a much higher value than for a given treatment, you can turn that into a node-edge node relationship between those concepts. So that's the kind of thing that you'll have to have that sort of pre-planned out of like what knowledge are you including in the knowledge graphs as opposed to just throwing data out there. And so yeah, so we're working right now on pathway figure OCR. So what that means is that not only will we be returning to you a result that shows you the name of a substance that might up or down manipulate a gene, but also a relevant pathway figure from one of the papers that is supported or that supports the data. And so then you can hopefully see very clear, right, from a very clear visual where that gene of interest or drug of interest might fit within that pathway. And so we're working on that right now. Eventually, we'd love to be able to generate new pathway figures based on the graphs that we're returning. We're far away from that, but that's something that we think we can also do. Okay, so let's stop there and ask, are there any questions about Translator before I start talking about LitCoin, which is my other program? Yes.
So when you get a list of, we'll say, chemicals and drugs from your query, is there any weight given to which ones come up first? Are those the best options for your query? Are they the ones that have the most literature support? Are they all equal?
It's a very big question. Yeah. So the question is, when we get a list of results back, what determines what placement within that list each of the given results are? And so the answer to that depends very strongly on which of the tools, the reasoning agent tools, sent that result back. So one of the things that we try to do from the instantiation of translators, we didn't want to have just a single way of answering a question, because context matters, methodology matters a lot. And so our 5 different reasoning agents have different methodologies that they use in the background to answer these questions. So right now, the way that we order the results is based on the scores that are coming directly from those arrays. They have a score range of 0 to 1. And the arrays that give us the, if you give us a 1.0 score, that means that reasoning agent thinks that that is the best possible answer for that given query, given their underlying reasoning that they put. But you can definitely have a case where 2 different reasoning agents return the same chemical as a potential upregulator, downregulator. One of them says it's a really great result. One of them says, there's not a lot of data for that result, right? We actually combine those 2 together, combine the scores in a way that I don't have a good grasp on, because that's being done by our very programming people. But we combine those scores together to get a sort of average score that then determines where it gets coalesced. And so, that's also based on the number of publications in a lot of cases. It can be based on whether there was a curated knowledge source, like a curated database that supports it, which is given a little bit more weight than some of our other metrics. It can also be predictions that were made by the underlying algorithms that these reasoning agents are used, and those predictions are weighted a little bit lower than the direct curated database entry support. So the answer to that, the overall answer to your question is it's complicated, and we're always improving. Like we're working on major improvements to that scoring algorithm right now that should be being updated. And I think we have a new release in February of the tool that we should see some improvements to that scoring algorithms.
Thank you.
Yeah, no problem. Other questions? Yeah.
Yeah, I understand some of your original data sources are actually not public. How do you handle that?
So we have, the question was, we have, when a data source is not public, how do we handle it? And the answer to that is, right now, all of our data sources are public, but the underlying data in some of them are based on actual clinical data from clinical records. And so in that case, we have these teams that have access to this large amount of clinical data. They have created some ways to de-identify the data and conglomerate patients together into summary statistics. And so the summary statistics themselves are what constitute the open data that we actually use and access in Translator. We don't have access to specific clinical records, but only those summary statistics from these large clinical databases.
Question, Ham's question, about conflict and variation in data sets. So there's a high-profile paper that we just heard about in this room a couple of days ago that shows that taurine decreases prominently with age in humans, and that boosting taurine is a good thing. But in other studies, like the Baltimore longitudinal study of aging, it's flat.
Yeah.
So in addition to weighting the scores, is there a prospect in the future of providing a controversy or a variability?
Yes, that's a really good question. So the question was basically, how do we handle both the case where there's contradiction in the underlying data, but also is there plans in the future for creating sort of controversy score or something to indicate to the user that this is something that is not a quote scientific fact or medical fact, but may have controversy associated with it based on different types of data. So right now, the way we handle that is by returning all of that data to the user in the same graph, in sort of branching paths, and representing it to the user and letting them dive into the underlying knowledge sources and why that controversy exists. Why are there 2 paths, one that says it upregulates and the other says it downregulates? In a lot of cases, that is dependent on the context of the paper that was used to publish it or something like that. It might be in 2 different body systems, it might be a blood-brain barrier question. All those things can lead to these sort of results that are, not matching up between different sources. But we do have a lot of research within the consortium going on right now on what would constitute a result that is new or so what's the term they like to use for this? Basically something that represents a difference from previous understanding. Novelty, sorry, they're working on a novelty score, right? That's a very complex question of how do you rank the novelty of a given result. You know, you could say that a result is novel if it's supported by one brand new paper and you know, that isn't supported or refuted by 5 papers from 20 years ago, but it might be that that brand new paper is crap. It might not be, you know, useful. And so that's why we want to make sure we're showing the user all of those underlying things. But that novelty score, I think, has a purpose. And a lot of cases, especially when we're talking about rare diseases, the patients have, you know, their clinicians have tried everything already, everything under the sun that they could think of for treating their disorder. And so they're looking for things that are what we like to call unknown knowns or underground truths, where there is data that supports something, but it's so hidden in the literature or hidden in a database that few people have access to or something like that, that the clinicians don't often have access to it, if at all. And so we have a really great example of that. This is from, this is way back before COVID, actually. This was the case where we had a young woman who had cyclic vomiting syndrome. So this meant that this young woman every single day was getting sick and vomiting for seemingly no reason. She just would vomit multiple times a day, and it was very horrible. As a 19-year-old girl, she weighed 75 pounds when she was seen. And they had tried everything, all of the common treatments for nausea and vomiting, nothing was working for her. And so her doctor came to Matt Might at UMB and said, we need to find something, what do we do? And so they did a search through Translator and found one publication from, I think it was 15, maybe 18 years ago, that said inhaled isopropyl alcohol can reduce nausea. And you know, when they saw this, they were like, well, that seems like something you can try at least, right? It's just isopropyl alcohol, let's try it. And so the doctor gave the girl a vial of isopropyl alcohol with some cotton balls in it. And every time she started to feel nauseous, she would open it up and sniff it. No nausea. 6 months later, she hadn't had a single vomiting episode. And this is the case where there wasn't a lot of literature about it because it was something that nurses have known for decades. Nurses all the time will hand a patient a little vial of isopropyl alcohol and start feeling nauseous and say, sniff this. But it was something that was not well known in the doctor's minds, right? This is something, and that's why we like to call that an underground truth. Something that was known, but not by the right people. And we had to connect the data in the right way to actually get it in the hands of the clinician, right? So that, I love talking about that example, because that, you know, it literally saved that girl's life. It changed her life completely. And it was as simple as finding the right paper from many years ago and getting it in the hands of the clinician. Cool. All right, so I'm going to jump to my other program, which is called LitCoin. So, what is LitCoin? Why do we call it LitCoin? So LitCoin, we like to think of it as a new publication format. And I'll explain that a little bit, but it also involves several layers of technology that we want to build into it. But we're thinking of it as a new publication format, which is citable and indexed by PubMed. And the goal here is to create something that is a new, smaller, sort of bite-size currency of research. So right now, our currency for your research, for your career, is how many papers have you published, and what is the, you know, your index number, right? What is the impact you've had, the impact factor in each of those publications, right? And so we want to create something that is easier to build, easier to publish, and computationally accessible from day one. So that's what LitCoin is meant to do. So we want to incentivize data and knowledge sharing through this, and we want to build AI-ready knowledge from the free text that you normally be publishing at the inception rather than post hoc. And so this will encourage researchers to share their data in a computationally accessible fashion. It's going to generate highly connected knowledge networks from published works with built-in curation by the authors themselves, but without undue burden to the authors. And it will encourage the reporting of research findings that might be independent of your main hypothesis that you're working on your next big paper. So this is essentially what we're doing right now. And this is funded by the HEAL initiative at NIH, Helping End Addiction Long-Term Initiative. And so we're piloting this LitCoin project on pain and addiction research. And so we're going to be generating human readable knowledge. And I'm using one of my own papers here, the abstract from my own paper, just to represent what the natural language processing is doing in the behind the scenes of LitCoin. And so you might take the text that's shown there on the left, and a natural language processing algorithm would pull out all the highlighted pieces, and it might create this type of graph right here. This is a machine readable knowledge graph that represents the knowledge that's found in that abstract. And so, for example, these, you have from one causes congenital diaphragmatic hernia is a simple knowledge assertion, but there are these contexts there as well. So the gray pieces here represent the context of that assertion. So in humans and mice, in this case. And from one is an allele of the from one eyes two is an allele from one in mice. So mouse allele. So those kinds of contexts, contextual pieces of information are really important. And we want to be able to create an algorithm that can pull out not just the high level knowledge assertions, but also those contextual relationships. Jumping forward. Sorry, there's a couple other things here. So the goal here is to take this entire thing. So the idea is you upload this free text, publication information, what you would normally be publishing in a journal. You upload that to our server. Our server uses natural language processing and LLMs to create this knowledge graph. And then this knowledge graph and the text that go along with it are packaged together into a submission package. That submission package then is sent directly to one of our publishers that we're going to be working with. The publisher then takes that submission package and sends it out to a bunch of reviewers, just like they normally would right now with any other paper. Those reviewers look over not only the text itself, but also the knowledge assertions that came from that text and say, yes, that is supported by the data that is in this package, right? It's supported by the data. Then the publisher gives us an okay and says, we're going to publish this. We haven't made, our reviewers didn't see any major problems with it, so no change need to be made. And they send it back to our LitCoin server, a little thumbs up that says we're going to publish it. And then we take all those new assertions that were generated and put them into our knowledge graph that we're building to add new associations and new relationships within our knowledge graph. And so in that way, we will continue to build out this LitCoin knowledge graph with new research every time a paper is submitted. Yes.
So Tyler, the way you describe the edges here, it's primarily as an indicator pin. In other words, it might be a -1, a 0, or 1. It's not a quantitative scale usually, right?
Right. Yes, these are definitely.
Limits gets a -1. So other words like increases get a +1. What can you do to capture? So the type of data a lot of us here make is highly quantitative, so it would be an R score.
Right.
So could you in principle capture that kind of data?
Yes, absolutely. So the question is, can we add quantitative data to this and not just qualitative assertions in the form of graph? And yes, so often the way that we expect to do that will be by adding the context to the properties of the nodes and edges themselves. So in addition to whatever this might say, FRAS1 causes congenital diaphragmatic hernia, you're going to have those contextual relationships in the properties that are attached to a given edge. And those properties can be anything from, this occurs in mice to with a p-value of 0.1, right? And those are all things that you can include on those edges that then can be computed across. We actually have, right now we have a young man who's started working with me. He's just got his PhD from Cambridge, and he's an ODSS data scholar, so he's going to be with us for 1 year working on this project. And the first thing he said, because he's very interested in clinical data, first thing he said is, how do I include p-values and hazard ratios and things like that? And I was like, well, then we're going to have to build those into the edge properties of each of these edges. Yes.
So a related question, if there is a confirmed no relationship, is this captured or is this just absent? Like, this paper show that there's no relationship between these two.
Known relationship. So when you say, this is something that was published 30 years ago, and it's
No,
or
and no and no.
there is a paper said there is no relationship between X and Y. Yes. So that would generate an edge for this graph, and the edge property on it would be, no relationship or no statistical significance, right? And so when you're computing across edges like that, that has to be taken into account. That's a special type of edge, because you're not going to use that as positive evidence, right? But you're also not going to use it to contradict positive evidence that comes later. So yes, we expect to have edges that say there was no relationship between these things, but those can essentially, in our headcanon for this, they can be overridden by positive relationship data that comes later. Does that make sense? Yes.
How do you protect against fraud?
How are we protecting against fraud? So the first way I would say is that's why we still want to have review built into this.
Sure.
The second answer is we can't always, right? And I think that's something that we have to accept especially when you're dealing with things like trying to get NLP on published papers. There are going to be a certain percentage of papers that are not great. Of course, if we see so one of the things we'll have to do is, if there's a retraction, then we have to be able to connect, to the paper and the retraction and say, all right, we need to remove those edges from the graph, right? That's an important piece. Anything that's retracted, it should not generate an edge to be included in our graphs, right?
So you have a situation where the graph just disappears?
You could potentially have a situation where certain edges within the graph could disappear, but it's, that's based on the idea that retraction had to be issued because there was fundamental flaws in the paper, right? So that, I think that's something that we as researchers just have to we have to accept that there's not always going to be a perfect representation of knowledge in the papers that are being published.
I'm editor-in-chief of the journal, and over the past few months, we've had a big increase in the number of claims of fraud already. I mean, some of it's relatively new. So it seems like you have to protect yourself here as well.
Yeah, and I think one of the, right now our beginning, to upfront the way that we want to protect ourselves is we're not going to put edges in the graph until the publisher says, yes, we're going to publish that paper, right? That's our first thing. And that uses the peer review process, and there are positives and negatives to the peer review process, no question. But it will give our users of this eventual system a bit of confidence that the underlying data, the underlying papers that are being used for this have at least been vetted by the peer review process. So I think that's where we have to start. I would love to say, eventually we want to start doing this for things on like bioarchive and things like that, and we have to have some sort of, a little asterisk that says this is based on data that was published but not peer reviewed. Like I think those are the kind of caveats we have to build into this kind of system. Okay, so go ahead.
Yeah, could you actually reword and rate and reword like how shareable is the text? Like, and from one side and from the other, kind of like this also require education for
Yes.
people who write it,
Yeah,
make it.
so
Which
you're
I
asking,
think it's great.
yeah, so you're asking about the education of the researchers on how their text is interpreted by.
Because if I understand, if I see where you want to go, you're probably going to reward people who make papers which are shareable, which is great.
I mean, yeah, so to start with, the reward for making papers that are shareable will be, more more published small papers, right? More published, more DOIs to attach to your name, and those being connected to you as, through your work ID or whatever, right? That's the short term. But in the longer term, we would also like to try to build in a reward system for being the reviewers on these papers. So if you are willing to review, LitCoins, then we feel like there should be a reward system for people who are actually able to put the effort into. So that's definitely something we're thinking about. I don't have a good sense yet on exactly how we want to do that. But when you're tracking people's work like this through these knowledge graphs, it would be very easy to also track who was reviewing in the, and somehow, add a reward system to it. Now, I will say the name LitCoin often makes people think, you are creating an actual, the first question is, are you making a currency that will actually be given to these people that is worth real world dollars? No, we don't want to do that. We named it LitCoin because we feel like it's the currency of the publication that we want to change, not actual real world money. Let me go forward here. So, so for this program, we had two prize challenges that we've the first one was focused on the natural language processing for the system. So we actually worked with the NASA Tournament Lab to design a challenge where the teams, I think they had something around 50 submissions from different teams, and their job was to run and create and run an NLP algorithm on a set of hand annotated abstracts. So we had hand annotations and we had their natural language processing annotated abstracts. And then we had a software system that compared directly how close was their NLP to getting the same output as the hand annotated abstracts. And they were ranked, and it was an iterative process, so they could see what their score was at a given moment, and they could go make improvements to the algorithm and come back and run it again and get a better score. And so this was an iteration over about, I think, like 2 months. And we ended up with 8 winning teams, eight prizes that we gave out for this, total of $100,000 in prizes. 3 of those teams had no academic or corporate affiliation. So they were individuals participating in this challenge, which is something that NIH normally doesn't get. We don't get the ability to give money to individuals working on a research project, right? And so that's one of the reasons I really like using this challenge, this prize challenge mechanism. And so we were granted a broad license to reuse the 8 NLP systems from those 8 winners for our LitCoin projects. And so we have 8 systems now that we're going to be picking and choosing the best parts of to build the actual natural language processing system that LitCoin is going to use. And so I'm going to skip past this. So I want to shout out Dr. Jiang Liu at NCBI, who gave us the initial hand annotated set of abstracts. And so we used that during our challenge to determine the winners of the project. This is where our winning teams were located. So you can see a lot of them obviously in the US, but we also had a group that had a sub award in Penza, Russia, and Chennai, India, and we had winning teams in the Netherlands and in Portugal. So this was a worldwide event, and another thing that really is cool about this is that we were able to give direct prizes to these people from these other countries, which you normally can't give a direct grant to an international awardee. They have to go through a sub award from a US based place. So it's an interesting mechanism. I'm really proud that we were able to use here. I'm going to skip the objective slide. So we think that this really has a lot of potential for building computationally accessible knowledge in the publication process that we're already using, and really incorporating the knowledge sharing into the systems we're already accessing right now. We think that it will lead to findings that haven't been published at all being accessible in a functional way. So, right now we have a lot of data that you're generating in your lab that's just sitting on a thumb drive or in a lab notebook, that unless it contributes to the story that you're telling for that next big paper, it's going to continue sitting in that lab notebook or on that thumb drive. But when, as you're generating this data, if you know that, that result isn't really supportive of the next thing that I'm working on for this big paper, but it's still valid data, it's still, valid research. Throw it into a LitCoin, write 2 or 3 paragraphs about it and attach the data and submit it to our LitCoin server and have it published as a LitCoin micro publication, basically. And that will get that information out there in a computationally accessible way so that other people won't try that same experiment and have it fail the same way, right? So that's one of the things we think we can do with this. It could lead to publishing failed drug discovery attempts. This happens all the time right now, where drug companies are, spending tons and tons of time and money on new discoveries in a given, druggable treatment area, and they might each try the same thing, a whole bunch of times. If we can get those published, if we can get the drug companies to say, this didn't work for us, let's throw it up there and move on, right? If we can get that information out there, we can reduce the amount of wasted money on these kinds of problems. Yes.
But what would be the interest in publishing negative results? Like, you have to lure them to publish, otherwise they won't.
Yeah. So the question is, what's the lure to add to get people to publish this? Yeah. So I think
That's what I'm saying.
Mhm.
She kind of like.
no, I totally get it. So one of the things that I think is going to be a clear help is, I didn't mention it when I was going through this, but one of the steps of the submission process for LitCoin that we have planned is when you submit your text and get your knowledge assertions back, and you have your different types of knowledge assertions, we want to show you how often that same knowledge assertion or similar ones have shown up in previous literature. And then link you to those publications. So for early career investigators, for example, it could be a really good tool for building the references at the end of your upcoming paper, something like that. That's one thing that we think. But the other one really comes down to getting credit for work that traditionally has been difficult to publish. If you made a new piece of software that is going to have value in the research space, right now, it's still not super easy to publish those things. You have to end up building this huge story about how that software is going to, revolutionize everything, right? That's the only way to get published. And so we think that this could be also a way to get credit for these, sort of incremental steps in research that get skipped over or get pulled together into a giant paper that doesn't, has 500 authors, Yeah, so we think that we're well suited to run pilots of this kind of software to show the feasibility of these kinds of concepts. Right now, we are building the foundational knowledge graph. So this is an extant contract that we have with RTI International. So they're working on taking the free text from existing publications in the pain and addiction research space and generating that initial knowledge graph. But this is from already published work. And so the problem with generating knowledge from already published work is someone has to curate it, right? Someone has to go in and make sure, oh, the NLP got this wrong and that wrong and this wrong, right? And fix those things. So the reason that we're working with HEAL is that they have a group of investigators that are funded by HEAL. It's about 150 investigative teams. And they have yearly meetings where they all get together and show off their new research and do, they work together. And so our goal is to take the knowledge graph that we're going to be generating from all of their publications and take it to one of their meetings and say, hey, we're going to have a 2-hour hackathon. Go through each of the papers that were associated with you and check and see if those assertions that we generated are valid. And if they're not, just quickly give us a sentence what we got wrong so that we can retrain our algorithm. So we're going to make use of this really great consortium of researchers to give us the initial curation of their papers that were already published. And then in the future, as we're adding LitCoin into the publication pipeline, researchers will be curating their own research right on the spot. They're going to be shown those new assertions and they're going to confirm, yes, this was right or no, this was wrong. Yes, so I think that is it. We have, yes, future plans for LitCoin include right now we're piloting the program to build the initial knowledge graph. We also plan to do a user interface for this in the, near foreseeable future, where researchers will actually bring their own, new research and upload it and get the knowledge, assertions. Then we want to expand to other fields. So, our pilot is in the pain and addiction research space because we're working with the HEAL investigators, but we definitely feel like this is something that we can expand into all fields of biomedical research and potentially all fields of research outside of biomedicine. Because, when we were working with NASA on this, they immediately saw the potential for the research that they're doing in the space program and getting that information connected in, useful ways to other types of data. So that's sort of where we're going with that. I want to give a shout out to our teams, to our LitCoin team and Biomedical Data Translator team at NCATS. These are all the people that I work with every day to build Translator. I did mention Aaron here. He's our LitCoin data scholar. He's working with me on LitCoin. Everyone else is working on Translator. And then this is other acknowledgements of people who have helped with both of these programs. But also on this slide, you can see at the top is the URL for accessing Translator. So it's ui.transltr.io. You can go there right now and try out Translator, see what you think. Let me know if there are things that are good and bad about it. There's a feedback button on the Translator webpage where you can submit feedback about the results that you see. And we're always looking for users to do user testing on Translator, because this is, again, an alpha build right now. We have a lot of improvements we want to make to it. So that is my talk. Questions about LitCoin?